being here, spending time with me, listening to what I have to say. I hope it's interesting. If you have any questions, please feel free to ask them. And furthermore, I hope this will be just a broad overview of what we have and what you need to consider when working with RNA. But I think we will go more into details when you come and visit us in April, if everything goes well. So let's get started. Yeah, first of all, a disclaimer, of course, I will show some products. I don't want to emphasize on them because this is not a commercial talk, but actually I need to talk about QIAGEN products because they're the ones I know my way around with. So I just wanted to make sure that you know that all the products I show are just for molecular use only and not for diagnostic purposes. Anyhow, here is how the agenda looks like. I would like to divide the seminar today into 4 parts. First, I would like to talk a little bit about the RNA universe in the cell. What kind of RNAs are there? Then I would like to say something about general remarks when handling RNA, followed by stabilization of RNA, and then special considerations depending on the sample material you will use for your RNA isolation. Now starting with RNA types in the cell. Actually RNA, the name is of course ribonucleic acid. I guess you all know that. And it's not only in the cell, it's everywhere in the cell, but also outside of the cell in exosomes and also bound to proteins. And there it's used for communication between cells. There are different types of RNA. One is the messenger RNA, short mRNA, which is transcribed from DNA and then goes into the cytoplasm and serves as a template for synthesis of proteins. Then we have the ribosomal RNA called rRNA, which is present in the ribosomes and helps the protein synthesis. And also the transfer RNA, tRNA, is a player in protein synthesis. It delivers the amino acids to help to connect the amino acids to protein chains. The microRNA is a non-coding small RNA that mediates post-transcriptional gene regulation, and then we have the long-coding RNA, which is actually one of the newest things, but also already a few years old. It can be up to 100 kilobase pairs long in length, and it has diverse function in the cells like epigenetics, alternative splicing, and nuclear import. Some further data about this. I'm not going to talk about all of this because I think it's boring to see all the numbers, but I think what is fairly interesting is that a cell can contain different amounts of RNA. It's also depending how active the cell is, like a cancer cell has a lot of RNA, a fat cell probably doesn't have a lot of RNA content because it doesn't do much gene regulation and protein synthesis. But in general, you can say that one cell contains 1 to 30 picograms of RNA, and most of that RNA is actually rRNA. It makes up to 85% amount of the whole total RNA, and followed by tRNA, which is around or up to 20%, and mRNAs only make up to 5% content of the RNA. Okay, now to general handling remarks. Actually, RNA is not that easy to work with, but please don't be scared of that. But it's less stable than DNA, and this is actually due to RNases that are everywhere in the environment and can easily be introduced into your experimental setup. So generally, I would say work quickly but carefully. And one very important point is to always wear gloves and change them frequently. The best way to work with RNA is use disposable plasticware because this is sterile and does not contain any RNases, so you don't need to require any pre-treatment to inactivate them. If you don't have disposable plasticware but non-disposable plasticware, there are different ways to treat this. Here you can rinse it, for example, with 0.5 nano NaOH and 1 millimolar EDTA, followed by RNase-free water rinse, or you could rinse it with chloroform to inactivate RNase-free, but please only do that if the plastic is chloroform resistant. If you use glassware, you can clean it with a detergent, rinse it, and then bake it in the oven at 240 degrees for at least 2 hours. I know when I went to university, we always autoclaved RNAs for 2 hours. And actually, but this is not suitable because there are still RNases that can be active during that conditions. So autoclaving is not the right choice. Alternatively, you can do a DEPC treatment. Here you can fill the glassware with 0.1% DEPC in water, incubate overnight at 37 degrees, and then autoclave or heat to 100 degrees for 15 minutes. This will inactivate the DEPC. And then everything is fine. Okay, so these are just some general remarks to keep the environment clean when you're working with RNA, but actually people always consider the isolation process to be important for having good quality RNA, but actually it starts much before that because your RNA is at stage, your expression level, by the time point when you draw your sample. Because as soon as you draw your sample, some expression changes can take place in your sample material, leading to induction of genes or downregulation of some genes and even enzymatic degradation of mRNA, which then will change your expression pattern. And this is what you don't want when you want to look at RNA expression. So this is shown here, right? To overcome the stabilization of your sample material is of great importance. And here you can see some experimental results. On the left-hand side, we took cells which were unstabilized and stabilized, let them stand there for over a time course of 60 minutes. I mean, this is not what people normally do in the lab, but this is just to make the effect of unstabilized cells obvious. And what you can see here is in the upper part of the picture that the ribosomal RNA is already or shows already degradation after 10 minutes and is really in a bad shape after 15 minutes. If you look at the stabilized cells, you can see that the RNA integrity looks very good over the whole time period. At the lower part of the picture, you can see another blot of the GAPDH gene. And again, here, the results is in accordance to the ribosomal RNA on the gel. After 5 minutes or 10 minutes, you can see slight degradation and no gene is left after 15 minutes. This is not true for the stabilized cells. Here you can detect the gene also after 60 minutes of storage. On the right-hand side, you can see actually the same results just with saliva samples. Here you can see with the blue squared bars that show the stabilized saliva samples from 3 different donors, and you can see that the expression level doesn't change over the time course of 72 hours. This is not true for the unstabilized material. So here, the gene regulation or the RNA is degraded and downregulated, and the CT values go up. So actually, now you can see some QIAGEN products, but be aware there are also products from other companies in the field that help you to stabilize your sample material. What I wanted to point out is actually that there are different stabilizing reagents for different sample materials, and I get a lot of feedback from the field where people just have stabilizing reagent, for example, from tissue samples and would like to use it for cells or saliva. If you have the chance, I would recommend you stick with the appropriate stabilizing reagent, because this is really doing the trick for your sample material, and don't mix up a different stabilizing reagent with different sample materials. Another point that I would like to make is actually, if you don't want to buy a stabilizing reagent, Using liquid nitrogen is also a very good tool to stabilize your samples if you freeze your samples right after sample extraction, and then you can use them and thaw them right before RNA extraction. You always have to make sure that there is a time window when you do your sample extraction or right before your isolation and disruption, where expression can be affected, right? Because then the sample is thawed, and if the sample is thawed, then gene expression can already take place. So you have to keep that time really short and work really fast. Okay, now we have the stabilizing in place. All the lab is clean and no RNases are around. Then you need to consider, depending on your sample material, some aspects how to get good RNA quality. Here, first of all, a general workflow for an RNA isolation. This is actually true for most commercially available kits. So normally, lysis is done in a chaotropic salt buffer because the chaotropic salt will help to denature proteins, including RNases and DNases. And after you have done your lysis appropriately, you will do the binding to the silica membrane. By the way, this is my son. Yeah, sorry. And yeah. Anyway, so you will bind the RNA to the silica membrane, and here the chaotropic salt is also of help. As you can see at the right-hand side, that the chaotropic salt kind of steals all the water in your lysate, and so the DNA or the RNA is kind of naked and then is forced to bind to the silica membrane. So actually, the chaotropic salt is important for the binding of your nucleic acid to silica. This binding is also done in the presence of alcohol if you talk about RNA isolation. So you need alcohol, and the concentration will be higher the smaller the RNA is you want to isolate. So for miRNA isolation, you will need a very high alcohol concentration in your binding. After you do the binding to the silica membrane, you will start to wash away contaminants by a first wash, which is also containing chaotropic salts. And then you will start to wash away the chaotropic salt by an alcohol wash, because chaotropic salts can interfere with downstream. At this point, I would like to point out that in the past, downstreams have been, they reacted on chaotropic salt concentrate or contaminations in your elute. But in the last few years, companies have developed very stable downstream assays so that chaotropic salt contamination is not that that problematic anymore. Still, people would like to get rid of this because it will interfere with OD measurement and can kind of raise your RNA yield, even though it's not as high as it's supposed to be. So anyhow, ethanol will help to get rid of your chaotropic salt on your membrane. And after that, we would like to get rid of the ethanol, because this can also have an influence in your downstream analysis. And we do this by drying the membrane, mostly done by a centrifugation step. When the membrane is dried, you can elute the RNA from the membrane using water or low salt buffer, mostly at a neutral pH. So this is actually the normal recipe to isolate RNA, but now we come to special considerations when using different kind of sample material, which has to be done, for example, upfront of your RNA isolation. And one thing is an efficient sample disruption and homogenization. So why do you need an efficient disruption and homogenization? You need it because it will properly release the nucleic acids from the cell before purification. This will then maximize your RNA yields. You can imagine if your cell is not open, RNA cannot bind to your membrane, but if you crack the cells open, of course, then the RNA content is released into your lysate, and then the RNA is available for binding to the membrane. Furthermore, as I said before, the chaotropic salt will help to avoid nucleic acid degradation because it can inactivate endogenous DNases and RNases. So if disruption is not done fast, RNases might have a chance to degrade your RNA. And if you do the disruption, sometimes you also need to have homogenization due to high molecular weight components in your lysate, which will then interfere with the binding also. So you need to shear this high molecular weight components, and this is done by homogenization. So as you might have noticed, we differentiate between disruption and homogenization. The disruption is the complete destruction of tissue structure, cell walls, and plasma membranes. And this helps to release all the RNA in the sample. And you need different disruption method for different sample materials. For example, if you think about bone samples or teeth or maybe plant roots, you can imagine then that it will be very hard to lyse them just by adding a lysis buffer and using a bead mill disruption method. Here you would probably rather work with liquid nitrogen and then maybe a mortar pestle or something that is more able to crack hard tissue samples. On the other hand, if you use cells, you can have maybe even just pipetting up and down or vortexing with glass beads, and that will help you to have a good homogenization in place. Disruption, sorry. The homogenization will reduce the viscosity, and this is due to the high molecular weight components like DNA in the sample. If you have these high molecular weight components, they will lead to a very viscous lysate, which is very hard to handle. And of course, it will interfere with binding of the RNA and therefore lead to reduced yields. Here you can see some effect on the recovery of RNA on a qPCR or a real-time PCR experiment. On the left-hand side, you can see that you get almost no gene expression or no PCR product if you do no homogenization, and same is true if you just vortex here. If you use something like the cryoshredder, which is a column-based disruption and homogenization method, which can be used for cells, for example, you can see that your yields go up, and same is true for an electric homogenizer like a rotastator or a bead mill-based disruption method. Sometimes a mechanical disruption is not enough. Now, if you're thinking about very difficult tissues like heart and muscle or other fibrous tissue, because they contain a lot of contractile proteins and connective tissue, collagen, and these can interfere with isolation process. So it's not enough just to have a mechanical disruption, but here we would recommend to treat these fibrous tissue either with a protease or a phenol-containing lysis reagent. Actually, if you don't mind working with phenol chloroform, this is a good solution for a lot of problems that you have, because phenol is not only nasty, but it's really good in lysis and in inactivating and denaturing proteins. And then you have the phase separation, which will also already kind of clean your lysate before going onto the column and then having a real good RNA isolation procedure. But I know a lot of people don't like to work with phenol and chloroform, and therefore maybe a proteinase digestion step is also an alternative. Here you have to think about that, of course, you need an environment where the enzyme can still work. So if you have higher chaotropic salt conditions, of course, the protease will not be able to do its work properly. On the other hand, you need to have conditions that keep the RNases still inactivated. So that's kind of hard to find the right conditions for your protease digestion. Another type of material is, of course, blood. Blood, here you might know that it contains only small amounts of RNA, and the RNA quality is actually, it's hard to get high quality RNA because there is a lot of RNases present in blood. Blood contains not only cellular RNA from the cells in the blood, but also exosomal RNA. But it also contains a lot of contaminants which can interfere with the downstream analysis. These include like anticoagulants like heparin and EDTA. So if you have a choice, EDTA works actually fairly good, but heparin is killing your RNA isolation. So don't work with heparin blood tubes. One of my favorites is actually the FFPE, or the RNA isolation from FFPE. And this is because it's difficult not only in isolation of RNA, but also you really have to consider the sample preparation, meaning the production of the FFPE blocks. And most of the time people get the FFPE samples from a pathology lab. So people in this lab don't care about how the samples were treated before, but for your RNA isolation, it is essential that the formalin fixation is not done for too long. So if you use it for more than 24 hours, you will have cross-linking of proteins, RNA and DNA, so they're kind of stuck and glued together, and you won't be able to separate them. So your downstream analysis will be really compromised by longer fixation time with formalin. Furthermore, the quality of the paraffin used is also, or does also have an influence. So here you should talk about the pathologist not to use any additives and avoid any staining if that is possible. Another aspect is the storage of the FFPE sample. Normally the pathologists just store them at room temperature, but of course this will also compromise RNA integrity. So they should be stored at 4 degrees if you have anything to say here. If that is done and the blocks are stored at 4 degrees, then actually RNA is intact for up to 1 year. And now to the isolation process. Since you have now the paraffin embedded tissue, you need an appropriate deparaffinization step up front, which will remove the paraffin from your sample. And then you need to do the decross-linking to kind of separate the RNA from the DNA and the protein. Here, normally it's a heating step, which is more or less long, depending on the kit used. And actually, I think it's important to know that this heating step will always compromise your RNA integrity. So you will always have some RNA degradation when you try to isolate FFPE, RNA from FFPE samples. But the longer and the higher temperature you use, the better you will have the decross-linking. So the more RNA you will be able to isolate in a good shape without being cross-linked to proteins and DNA. So it's always a question how much degradation you can endure and how good the cross-linking must be. And then we can still come back to the 24 hours of formalin fixation. If this time is really short, of course, the cross-linking can be done in a shorter time period. Another very challenging sample material is actually the isolation from RNA from microbiome samples like stool. This can also be interesting for diabetes research, I suppose. And why are these so challenging? Because it is very difficult to have a lysis which will be able to lyse all microbiomes in the sample, because there is an extreme diversity of bacteria and other species in them. If you don't lyse them all equally, you will have a bias towards the easiest to lyse species, so your results are compromised. Also, there are several microbial metabolites that are released during the lysis that mask the PCR signals. So actually you have inhibitory effects in your downstream that you need to get rid of before. And also you need high yields, because you want to detect not only the high abundant species, but also the low abundant. And the effect you can see from the sample preparation is shown on the left-hand side. Here you can see different stool samples on the upper or the lower picture, and both show the changing over a period of a certain time. So you can see that even storage of your sample material might lead to a bias in the microbial content of your sample. So that's also very difficult to have here something in place that doesn't compromise your RNA isolation process. Now coming back, I talked about different input materials and how they can, how you need to treat them to get good RNA out of your sample. But something that is also generally important is the removal of GDNA during RNA isolation, because traces amounts of GDNA in your RNA samples can compromise the accuracy of sensitive applications like real-time RT-PCR, meaning that both targets, RNA and DNA, may be amplified, and then your CT values will be falsified. You can, of course, eliminate the GDNA contaminations either by during the RNA purification. This can be done either enzymatically using a DNase digestion step, or by a column-based GDNA removal using a GDNA binding column, or you can do the GDNA removal just prior cDNA synthesis. One other possibility to get rid of GDNA or the effect of GDNA in your sample is to design your primers so that they don't detect DNA. Okay, with this I come to an end, and as I said before, this is, it's very difficult to narrow down what to say, so I hope we will have a good time in April and maybe discuss some questions there. And maybe if you have any issues with your RNA isolation, you can also bring your samples in April, and then we might be able to see what we can do about that. But of course, if you have any questions now, I would be happy to answer them.